In the 22nd century, we don't just read DNA, we edit it, sculpt it, and even program it. But to understand how far we've come, we must travel back to the 21st century, a time when the double helix was only beginning to reveal its mysteries. In the 20th century, the discovery of DNA's double helix structure by Watson and Crick marked the birth of molecular genetics. By the 21st century, we knew that DNA, deoxyribonucleic acid, carried the genetic blueprint of life in 4 bases: adenine, thymine, cytosine, and guanine. But in the 22nd century, we now recognize that DNA is more than just a static code. It's a quantum-responsive, dynamic field of biological potential. The central dogma of molecular biology, DNA makes RNA makes protein, was once considered the foundation. But today, we understand it's more like an orchestra, with feedback loops, non-coding RNAs, and epigenetic signals playing conductor. In the 22nd century, we've discovered quantum entanglement-like behavior in biomolecules. For instance, the folding of proteins is not just biochemical, it's influenced by quantum coherence. That's why diseases like Alzheimer's are now treated not just with drugs, but with vibrational quantum harmonizers that correct misfolded protein pathways. DNA replication in the 21st century was understood as semi-conservative, each strand serving as a template. Enzymes like DNA polymerase, helicase, and ligase ensured this miraculous copying process. In today's world, we've upgraded to quantum-assisted replication units. Our synthetic polymerases have error rates so low, they're measured per trillion base pairs. And if there's a mutation, CRISPR-13 can edit it mid-replication using photon-sensing triggers. No need for invasive procedures. Earlier, transcription meant converting DNA to RNA, then translation into protein. It was all molecular machinery. Today, transcription can be neuro-driven. Our thoughts can initiate synthetic transcription, creating hormones, enzymes, or even neurotransmitters on demand. This is powered by quantum neural interfaces that interact with DNA's minor grooves and promote targeted transcription using guided light pulses. Mutations used to be feared. Silent, missense, nonsense, or frameshift mutations could cause deadly diseases. In today's world, we've turned mutations into tools. Want better lung efficiency on Mars? Or resistance to Arctic cold? A base pair tweak and it's done. But we also regulate this tightly. The Bioethical Genome Act of 2174 ensures no human editing happens without full ecological and psychological modeling. The genetic code was once considered universal and immutable, but we've now expanded it. With synthetic bases like X and Y, we've created new amino acids, leading to new materials, medicines, and even sentient biomachines. Imagine proteins that heal themselves when damaged, made possible by these artificial expansions of the genetic code. In the 22nd century, every citizen carries a quantum genome ring, a wearable that reads your DNA, predicts disease onset, and triggers protein therapies before symptoms even begin. Rice plants now express stress response genes from tardigrades, allowing growth in deserts. This was unimaginable in the 21st century when we relied on traditional breeding and early GM crops. Children don't just learn biology, they experience their own molecular history through immersive genome theaters, where their ancestral epigenetic stories unfold as interactive documentaries. Molecular genetics began as the study of a molecule, but today, it is the language of life, the script of identity, and the canvas of evolution. The 22nd century hasn't replaced biology. It has reconnected us with it, in deeper, more intimate ways than ever imagined. And to think, it all started with a humble helix in a 20th century lab.